Hello, everyone. It's great to meet you all today. I'm Mohit, and I have a PhD in computational biology and specialization in molecular modeling, simulations, and discovery related research projects. So, in today's session, in a few slides, I'll go through the publicly available data, recent structural data from the pandemic. So, talking about that, the structural biology response to the COVID-19 pandemic has allowed the world to see the virus, the virus is attacking us in a structural perspective. So we can visualize, we can see the structure. So it has also aided in vaccine and therapeutic development and has shown foresights in the investments in structural biology capabilities in techniques such as X-ray crystallography or techniques such as cryo-EM or the MX. So this sheer magnitude and uniqueness of the event that has enabled all of us in a way that helped in all these different types of data that has come out from different repositories. So today we are going to get an overview of where this data is and how it looks. So the speed with which these structural investigations have occurred has been really breathtaking. Simultaneously, the availability of these data has benefited from the long history of making these repositories and the metadata that is also available with this information about the structural information, and all of this is available in repositories such as Protein Data Bank, a worldwide repository for macromolecular structures. And also we are going to talk about some other aspects of getting structure, which is through molecular modeling. So as there is like this 1.5 years since the declaration of COVID-19, and recently 1,000 structures of SARS-CoV-2 biomolecules have been deposited in the PDB itself. So these are all experimental structures that have been deposited in Protein Data Bank. So it is like kind of rare to obtain a snapshot of such immense effort in structural biology, and this happened due to this revolution of not only the need or the structures, but also the technology. So with cryo-EM, there has been a lot of insights that we got from the resolution revolution. That is what it is said in aspect of cryo-EM microscopy. Talking about the computational aspect of this. So first, the experimental aspect is about the structural biology events and experiments that led to the information of this kind of data, that how these structures are. But still, there are many limitations when we are talking about the complete understanding of how structure can be related to the function. So that's where computational structural biology or computational structural bioinformatics comes in. So where we are talking about modeling complex structures to gain insight, or to be able to understand the effect of mutations on these experimental structures. So those things can be modeled and have been playing a very important role in understanding the pandemic more and more and to find solutions. So the SARS-CoV-2 pandemic, as I said, has contributed a lot in terms of also structural biology work. So in silico structural biology work. So initially what people were used to do is, I will tell you about some of the softwares that are available, online tools, such as the one that is listed here, AlphaFold. But also there is a software that many of us use for structural predictions is CI-TASR. So from Zen's lab. So it's an online available tool. And also AlphaFold from DeepMind Consortium. They have been able to with these tools, we have been able to leverage the release genomes, determine protein sequence, and also to create computational models. And this has been happening till the early March 2020 to till date. So some modeling, a lot of insights that we could draw from structural data is via modeling. So how do we model complex structures? How do you model single point mutations or multiple mutations and understand the variants? And also, then there is an aspect of molecular dynamic simulations that have massively also extended the utility of experimental structural data that helps in providing insights via the rapid analysis of these in silico mutagenesis for many key viral proteins, and also the ability to, for example, model glycosylation of the spike protein. Our computational approach has also helped in probing host pathogen protein interactions, so protein protein interactions, and then contributing also in network based and machine learning based analysis. And we have an upcoming talk about the network analysis tomorrow that I would like you to all know about Omics Club. So we will be sharing that information also with you. So about the structures and to understand like what is available out there and how to understand and make use of this. So that is an important aspect of integrating structural biology into your research, or utilizing the data that is available publicly. So the spike protein of this virus, I mean SARS-CoV-2, is one of the central figure, right, in this fight, since it is the primary target for antibodies to provide and to target of antibodies that provide immunity against the virus. So the surface of coronaviruses, I'll give you a layman explanation. So the surface of coronaviruses are covered with these spikes, giving them their distinctive clown-like appearances in the electron micrographs. So many people are already aware of this, and I think most of us are. So the spikes initiate the process of infection, binding to the receptor, and then fusing with the cell membrane to release the viral genome inside. That's the process. So many other, and it has been seen in many other models as well, like influenza, hematoglutinin, and also HIV and Ebola. So the spike protein is composed, of 3 identical chains that form together a complex with a small domain inside, which is inside the virus, which is the membrane spinning segment, and then there's a large ectodomain, and that all you can see some of those structures that you can see in this slide. So these structures also include a portion of the many sugar chain that coats the spike. Since the sugars are highly flexible and difficult to observe, that's one of the challenges. So many things that you can gain insight from are the structures, are from the structures. And to tell you more about that, so recent structures have revealed that the spike of SARS-CoV-2 and SARS-CoV and SARS-CoV-2. So comparing both, when we compare both, we found out that they are quite flexible. So in these structures, the receptor binding domains, the receptor binding domain are seen in different conformation. So when you compare structures and when you're superimposing them on one another in a 3-dimensional space, you can see the differences and how there is a movement. You can find out about that movement just from your screen. So by learning about these different tools that we will be covering in this entire program. So often just one domain, in this case of SARS-CoV-2. So many times what is happening is that one domain, you would see that there is some movement, and then there are several other extensions that you will see that they are tucked down, or I mean, there are different type of movements that have been found out in these different types of conformation. And then what researchers have hypothesized that the recent SARS-CoV-2 variants are virulent because their receptor binding domains are particularly flexible. So that's one of the hypotheses. And whereas most spread coronaviruses that cause the common cold are less dangerous because they are less flexible. So that's one of the hypotheses that you can understand by comparing some of these structures and understand from the different conformations. That include the binding. I mean, to understand this has to be modeled. So you have structures from different conformations that you obtain from different protein data banks, structures which are deposited online, and then you try to model them into a complex. That will give you a clear insight of how this is done. So these are some of the things that you need to learn computationally in computational biology or in silico computational structural biology. So about the receptor binding, our immune system, fight backs, right, when coronavirus infects us. So that's where the antibodies comes in. So antibodies are what? Are protein molecules that play a key role in any organism's defense against a foreign molecule called antigen, right? The first step in this process is specific complex formation between an antibody molecule and a molecule of the antigen. So the structural basis of this antibody antigen recognition is started to getting elucidated from these structures. So these structures are helping us to understand that mechanism, how it works. So the spike is a major target of this protection, right? Since it is exposed to the surface of the virus, and these three protein structures that you can see on the left, which is 3BGF and 2GHW and 6W41. And you can totally search them by their 4-digit keyword. You just paste them on your browser, it will show you to the link to their actual structure that has been deposited in the PDB. So these are PDB IDs. So these three structures show that the antibody can be recognized by spike protein in many ways. So two of these antibody block the receptor binding portion of the domain, but the other antibody targets a cryptic site at the base of the domain that is only exposed when the antibody binds to it. So you see that there are not only just different conformation, but different active sites also that you can find, that you can see, which depends on the movement of the protein. So one thing that you have to understand is that a PDB is just a snapshot. And when a protein is in motion, then it is moving. So there are different conformations that a protein attends. And then based on those different conformation, there could be different strategies to design molecule. So this is something that we should consider. These sort of structural information is something that we should consider when we go about, designing a vaccine, or when we are looking for strategies to find out solutions to, finding a drug. So about this, doing all of this is kind of possible. So in this program, we will be learning to use powerful visualization tools such as Chimera and explore through the available structural data. And then also we will be reviewing several tools developed for your training in this program to navigate large data sets, and then connect the various aspects of, sequence, structure, and metadata into a single experience. This is SeqNav. So what it will help us is that it will help us in understanding or addressing many questions. So genomic data analysis can be utilized to see the changes between viral strains. So you're looking at the mutations and different significance level of these detected changes, and how you track them or map them to the 3D position on their protein structure, so that you can see whether they are on the surface, whether they are in the active site, what could be the role. Then we will be able to do, from sequence to structure. Another aspect is to do comparison, and that comparison at a sequence level could be done through multiple sequence alignment. So multiple sequence alignment is a very powerful tool to understand viral adaptation between animals and protein, and again then trace it back to the structure. Similarly, another aspect of answering some of the research questions would be to understand the relationship between viral and host proteins, such as the trimeric SARS coronavirus surface spike glycoprotein. So this spike glycoprotein binds to a cellular receptor called angiotensin converting enzyme 2, so ACE2. I mean, you might all have heard about ACE2 in short. So, and it mediates through the fusion of viral and cellular membranes through a pre to post fusion conformation transitions. So as I was telling you before also, so because of certain binding, there are conformational changes that enables another binding event. So all of this fascinating stuff, and just imagine to be able to visualize it and to understand from sequence level to a structural level is something that we are planning for this program. And we are hoping that many of you can go through that experience and understand different aspects of how structure and complexes that mapping mutations from a single protein from a population to the three-dimensional structure and understanding the function obviously is something that we're trying to help everyone to learn. So this is what we have planned for the structural part, and that's what I wanted to tell you all about in today's session.